Hello everyone, you are welcome to my YouTube channel, and I'll be discussing the classical pathway of the complement system. What is the complement system? The complement system is a part of the innate immunity or non-specific immunity, which ensures that invading pathogens and abnormal self-derived components are eliminated. A complement system is made up of 30 serum proteins. These proteins are regarded as proenzymes or inactive, and when they are activated, they are involved in a cascade of biochemical reactions that leads to lysis of microbial cell or phagocytosis. Phagocytosis has been discussed in another video, and I'll encourage you to watch that video for full understanding of phagocytosis. There are three pathways of complement recognition and activation leading to effector functions of the activated components. These include 1, the classical pathway, 2, the alternative pathway, and 3, the lectin pathway. But in this video, the classical pathway of complement recognition and activation will be discussed. What triggers this cascade of events? The formation of antigen-antibody complex is what triggers the cascade of events in the classical pathway. The C1 component is made up of 3 proteins, C1q, C1r, and C1s. The C1 component is known as the recognition unit. And the C1q of this recognition unit binds to the Fc portion of the immunoglobulin, immunoglobulin G or IgM. The antibody-antigen immune complex bound to the C1q then activates the subunit C1r and subsequently C1s, which cleaves C4 into C4a and C4b. C4a is a small fragment, while C4b is the larger fragment, which combines with the activated C1 components. Next in line is the C2 component, which is also cleaved into C2a and C2b. The C2a is the small fragment, while the C2b is the larger fragment, which combines with the activated C1 components, C4b and C4b. Here is the diagrammatic representation of the activation of C4 and C2 components by C1. You can see the progression. The C1, which is made up of C1q, C1r, and C1s, activates C4, leading to the formation of C4a and C4b. C4b combines with the immune complex, followed by activation of C2 into C2a and C2b. C2b combines with the immune complex. So the latter, which is the C4b2b, is known as the active C3 convertase. It is known as C3 convertase because it cleaves C3 molecules into two fragments, C3a and C3b. The C3b has enzymatic activity and it combines with the immune complex, and it is known as the C5 convertase. That is, it is responsible for cleaving C5 into C5a and C5b. Here is the diagram showing the activation of C3 and C5 components. The active C3 convertase converts C3 into C3a and C3b, while C3bb, which is now the C5 convertase, converts C5 into C5a and C5b. Now, the C6, C7, C8, and C9 components will have to be activated by the C5 fragment, which has combined with the immune complex. This leads to the formation of the membrane attack complex. And once this is formed, a pore is made in the cell membrane of the pathogen and cell lysis occur. It is to be noted that only IgM and IgG can fix complement via the classical pathway. This is the diagram showing the activation and combination of C6, 7, 8, and 9 components by C5b, leading to the formation of the membrane attack complex and then formation of a pore where cytolysis occurs. Let's look at the functions of the complement components in the classical pathway. It's important to know that cell lysis and death occur following formation of membrane attack complex, and the important inflammatory effects also occur following the release of the complement fragments C3a and C5a. For example, C5a is known to send a signal for the migration of neutrophils. That is, it provokes chemotactic migration of neutrophils to the site of complement activation. These neutrophils are like additional support for the elimination of the pathogen. They make their way to the site of hyperemia, where in the presence of C5a, they attach to the endothelium and they are involved in phagocytosis. Mast cells also release their content, leading to increased blood flow to the hyperemic anatomic sites, and blood vessels dilate due to release histamine. Thank you for listening.